I'm Ann Knowlton, and I'm a professor of medicine and pharmacology at the University of California, Davis. And I have just finished presenting a talk today at the International Academy of Cardiology Annual Scientific Sessions in 2015 on aging, estrogen, cellular senescence, and inflammation. Now, the importance of this topic is that aging is something that virtually everyone is going to undergo. And aging is accompanied by inflammation and many changes. You would have no problem distinguishing an 80-year-old person from a 20-year-old person at great distance. But we understand very little about the aging process. Previously, we have done some in vivo studies using aged rats. And when we look at aged rats that we study females with and without, we overoptimize them and we replace half of them with estrogen. We find that there's significant dysfunction in the vasculature and that estrogen mitigates some of the changes, but that aging causes increased constriction, which is reminiscent of the fact that as people age, they get very bad hypertension often that is difficult to treat. So we wanted to understand more about this, but it's very hard to investigate mechanisms at the cell and molecular level, which we need to understand things better in the whole animal model. And so we have moved our studies to the study of human coronary artery endothelial cells, which are the little cells lining the blood vessel, and very important for regulation of blood vessel function. And human coronary artery endothelial cells are available commercially thanks to people who donate their bodies to science. And then at their time of death, their cells are harvested. And so we're able to study cells from different age people, and we're mostly focusing again on estrogen and female donors. And what we have found is that not surprisingly, we can take these cells and grow them in culture, and every time the plate fills up, you pass it to another plate. And it turns out that we can predict ahead of time how many passages it takes, based on our experience, to get to a state where we call senescence, where in senescence cells, they are metabolically active, but they don't perform normally. It's like having a shirker at work who does nothing. And they are larger and flatter, and sometimes they have little vacuoles in them. And they also can be stained with what's called, they have activity of senescent beta-galactosidase, and so they develop a blue color when you give them the substrate for this enzyme. So these cells, it turns out, when they get senescent, there's something that has been described called the Senesin-associated secretory phenotype for senescence cells, and our endothelial cells, when we get them out to senescence, they produce IL-6, IL-8, and MCP-1 when we screen them with a cytometric bead array that would screen for 12 different inflammatory cytokines. And the younger cells, which are called early passage, did not do that, and again they would have normal function. But if we put the younger cells and the senescence cells together, we get more production of inflammatory cytokines than we had with either alone. And estrogen can mitigate some of these changes. Now, endothelial cells, one of their jobs in the universe, in fact, their major job, is regulating our vasculature so that blood goes where we need it. I mean, when we're eating, we don't want all our blood to be in our feet or in our head because we need to be able to digest food, so we need more blood flow. So the endothelial cells, this is one of their main jobs. And so we put our endothelial cells together with vascular smooth muscle cells in a culture system where both sets of cells are in the dish together, but one is suspended in a little transwell so that they don't touch each other. So we can just see substances that communicate between the two. And we found that when we grow endothelial cells with vascular smooth muscle cells in culture this way, the early passage, the young ones, when they're there together, we see downregulation of the expression of endothelin, or release of endothelin by the endothelial cells, and endothelin causes vascular constriction. On the other hand, the senescent cells, when they're in that setting, they do not change their release of endothelin at all, and it remains the same. So this suggests that in becoming senescent and not functioning normally, they lose some of the signaling that goes back and forth, or crosstalk, that regulates these important functions. And remember that vasodilation and constriction is totally dependent on the presence of endothelial cells in order for it to happen. So we feel that there is evidence to support that these cells may have an important role in changes that develop with aging. Plus, their inflammatory output is going to go throughout the body and can have deleterious effects. And we think that it's important for us to do more studies on aging, because aging is basically the substrate for most of the diseases we see, such as coronary artery disease and heart failure. We hope with further studies to understand the role of cell senescence in these diseases better.